Herkese tekrardan merhaba arkadaşlar. Bugün yine final haftası olduğu için yeni bir edit yapamadım ve hep beraber ders çalışmaya oturalım diyorum. Çünkü finaller geliyor üniversiteler için. Yine liseler için de YKS illeti var. Hep beraber çalışalım o zaman. Bizim sanal derslerimiz bu şekilde oluyor. Hoca burada slide veriyor. Şuralarda da zaten hocayı dinleyebiliyorsunuz. O şekilde. Hadi şimdi başlayalım. Hocanın ses kaydı birazcık kötü. Bir önceki canlı yayınımda notlarımın görünmediğini falan söylemişsiniz. O yüzden şimdi birazcık notları büyük yapacağım. Kendim arka plana atacağım ve kırık ekranım hala düzelmedi. Şu an kırık ekranından da yayın yapabileceğimi fark ettim. Çünkü kamera orada duruyor. Bomboş ekrana bakıyorum ve kırık bir laptop ama neyse. Notları falan buradan alacağım. Hadi o zaman başlayalım. Umarım size de yardımcı olur. Kendimi kapatıyorum.
And here we observe the example of rabies virus, that it has a single molecule of RNA negative polarity as genome, and you observe how this virus enters in the host cell, and the negative strand will serve as proteins, and by the other side in the host cell, and the negative strand will serve as template to produce by one side the RNAs to be translated into proteins, and by the other side replicative intermediates. Anyway, one thing that is important to know in general is that RNA virus remains in the cytoplasm. They cannot use the replicative machinery of the host cell, and that's why they in general remain in the cytoplasm. The general rule is RNA genomes remain in the cytoplasm, DNA virus remain in the nucleus, with two exceptions that you should remember. By one side, one exception of DNA virus that remains in the cytoplasm is pox virus, and another exception that you have to remember, is RNA virus that goes to the nucleus, that is the influenza virus that we. And this is the influenza virus. You see here in this viral cycle how the segmented genome of influenza virus enters inside the nucleus, and there it is produced the transcription. Each segment serves as template to produce mRNAs that later on will be translated into in the cytoplasm. Genome of influenza virus enters inside the nucleus, and there it is produced the transcription. Each segment serves as template to produce mRNAs that later on will be translated inside the nucleus, and there it is produced the transcription. Each segment serves as template to produce mRNAs that later on will be translated into in the cytoplasm, and each segment is also serves as a template to produce replicative intermediates, positive strands that will serve as template to produce new copies of the genome. You see here how the genome, the 8 segments of the genome of influenza virus, each segment gives rise to one different mRNA that will genome of influenza virus, each segment gives rise to one different mRNA that will be translated into one protein. So in general, one segment gives rise to one protein, with the exception of the first segment that by framesheet give rise to two proteins, and the seventh and eighth segment that by alternative splicing to be translated into one protein. So in general, one segment give rise to one protein. Give rise to two proteins, and the seventh and eighth segment that by alternative splicing will give rise to two different mRNAs that will be translated each one into a different protein. will give rise to each one into a different protein. Cuando tenemos genomas segmentados, puede ocurrir que si hay when we have segmented genomes, one possibility is that if there's different strains that infect the same host cell, there could be a reassortment of segmented genomes. That means that some of the progeny could have segments from one strain and segment from the other, and then we could have new variants with different serotypes, and that give rise to the virus antigenic shift. That means a big variant against which we do not have the immune system ready. In the case of the Familia Arenaviridae responsible of the choriomeningitis, lymphocytic choriomeningitis, or hemorrhagic fevers, or Lassa fever, and also in the case of the Buñaviridae family responsible of Rift Valley fever or pulmonary syndrome, both families, Arenaviridae and Buñaviridae, they have ambiscent segments. Both of them, they have a segment that is negative strand that behaves as a negative strand virus, and an ambiscent segment that it is the one that we are going to try to understand now. In fact, here in this scheme we have the S segment, that is the ambiscent segment of the Arenaviridae family. The L segment is the one that is a negative strand. But we right now we are going to understand only this ambiscent S segment. In the upper part of the scheme, you observe this segment that, as it is ambiscent, it has two polarities at the same time, negative and positive polarity. So this genome, when it arrives inside the host cell, the negative polarity will serve as template to produce mRNA that will be translated into one protein, that it is the N protein. This N protein is a protein involved in the replication of the virus. So it will make possible that this genome is transformed into the complementary strand, where the positive part will be negative and the negative will be positive. This will be a replicative intermediate that will serve as template to produce, going again up in the scheme, to produce more copies of the genome. Okay, but the genome as replicative intermediate, it has a new part negative, the one that it was positive originally, and this new part negative will serve as template to produce an mRNA that will be translated into a new protein, and this one, it will be a structural protein, it's a glycoprotein. Okay, so as you can see in this scheme, some proteins are produced from the original genome, some others from the replicative intermediate and separately produce the non-structural proteins from the structural proteins, as we have observed in other viruses. En la diapositiva anterior vosotros me podríais decir, ¿y por qué no se genera? And you may ask, and how come in the previous slide, when it is produced the mRNA from the negative part, why it is not produced the whole molecule, and why it is not produced a huge mRNA with the whole segment? And the answer is in this slide, because you have a structure in the middle of this segment where the transcription is stopped, okay, and the mRNA just only goes till this part. You have a structure in the middle of this segment where the transcription is stopped, okay, and the mRNA just only goes till this part. But once the end protein it is produced, it acts as an anti-terminator, as in the case of the negative strand virus, acts as an anti-terminator and will read the whole segment and will produce the complementary strand of the whole segment. Okay, and this is why just by this structure we discriminate mRNA from only a single part of all the rest of the genome. Now we'll enter with more detail in the class of virus, single strand RNA, positive polarity, with reverse transcriptase activity. We only have to remember one family, the Retroviridae family, and two human pathogens, the human immunodeficiency virus, HIV, and the human T lymphotropic virus, HTLV. The strategy of retrovirus to produce mRNA and new copies of the genome is very strange, very complicated in fact. You have here the positive RNA, the genome, that instead of being translated directly into a protein, what it produces through the reverse transcriptase activity is a complementary DNA of negative strand that will serve as template to produce a double strand DNA. In this phase, it is called provirus as double strand DNA. And this double strand DNA the negative strand will give rise to mRNA that will be translated into proteins, and also the negative strand will give rise to more copies of the genome, but through this intermediate provirus. Here you have in this viral cycle of HIV
Artık öğrendim. Aşağısı İngilizce, yukarısı İspanyolca.
how the virus, the only activity that is done by the virus is the reverse transcription that happens at the beginning of the viral cycle. Once they have formed the double-strand DNA, and as a provirus, this double-strand DNA circularizes, enters inside the nucleus, and get inserted into the host cell genome. And once there, it happens like any other gene of the host cell. The own host cell forms from there mRNAs or positive strand RNAs that will be translated in the cytoplasm into polyproteins that will be cleaved subsequently into different viral proteins, or some other molecules of positive strand will be encapsulated into new viral particles, or some of these mRNA will be alternatively spliced into to form glycoproteins that will form part of the virus envelope. So you see that the only specific viral activity is the reverse transcription. The rest is done by the host cell. In fact, the whole genome has a single promoter and act under a single promoter action. Here you have in this in this viral cycle of HIV how the virus, the only activity that is done by the virus is the reverse transcription. And in fact, due to the insertion of retroviral genome in our own genome, we have an 8% of our genome that has retroviral origin. These are genomes or stretches of genomes, of viral genomes with mutations, so they are not able to produce virions, but there has been some retroviral activity, traces of retroviral activity that sometimes had made our own mRNA to be inserted inside our genome as pseudogenes, and this may have caused some diseases or not. And also some viruses, other viruses, just because this residual retroviral activity, retrotranscriptase activity, they have been also inserted in our genome. To the insertion of retroviral genome in our own genome, we have an 8% of our genome that has retroviral origin. Retrovirus, as they have diploid genome, they have the advantage that they are relatively resistant to UV and ionizing radiation that in normal conditions would produce mutations in the genome that do not allow the virus to survive. But if they have two copies of the genome, in the process of replication, the polymerase will make a copy that may be a hybrid of the two original strands, because the polymerase jumps from one strand to the other. And in this way, it can produce and hybrid replica that is a mixture of both strands and that avoids the mutations. And this is the last group of genomes that we are going to analyze in detail. That is the Hepatnaviridae family, with the main component being the hepatitis B virus. At the bottom of this image, we observe how does it looks the genome. It has double strands, circular double strand, but relaxed. The circular DNA is not closed, so it is in a relaxed way, and both strands, positive and negative, are open. You can observe that one of them has a protein at one side, and the other one has a piece of RNA that it is believed that it is in a relaxed way, and both strands, positive and negative, are open. You can observe that one of them has a protein at one side, and the other one has a piece of RNA that it is believed that is the primer that the reverse transcriptase used to produce the double-strand DNA. That it is believed that is the primer that the reverse transcriptase used to produce the double-strand DNA, and the positive strand, it is not closed, it is quite open, and it is believed because it ran out of nucleotide triphosphate before finishing the complete strand. Out of nucleotide triphosphate before finishing the At the upper part, you observe the strategy of this hepatnavirus to produce mRNA and more copies of the genome. they first of all, what they do is just they complete the double strand DNA, and from there the negative strand serves as template to produce mRNAs that will be translated into proteins. What they do is just they complete the double strand DNA, and from there the negative strand serves as template to produce mRNAs that will be translated into proteins, but also to produce a copy of the genome in form of replicative intermediate as positive strand RNA, that it is called the pregenome of the genome in form of serves as template to produce mRNAs that will be translated into proteins, but also to produce a copy of the genome in form of replicative intermediate as positive strand RNA, that it is called the pregenome. And this will serve as template for the reverse transcriptase to build the complementary strand in DNA, negative strand, and from there to produce the double strand DNA. Here you can observe the viral cycle of hepatitis B virus and how when it enters inside the host cell, it goes the genome to the nucleus, and then there the double strand of DNA is repaired. It is complete and circular, so it is supercoiled, and it is repaired by the own polymerase of the virus. And then There, the double strand, the RNA polymerase dependent on DNA is repaired by the own polymerase of the virus, and from there, the RNA polymerase dependent on DNA of the host cell will produce the pregenome of RNA that you there. The RNA polymerase dependent on DNA polymerase dependent on DNA of the host cell will produce the pregenome of the host cell will produce the pregenome of RNA that you see in green, and also of course will produce the mRNAs that will go to the cytoplasm and will be translated into proteins. DNA of the host cell will produce the pregenome of RNA that you see in green, and also of course will produce the mRNAs that will go to the cytoplasm of the capsid are produced and into proteins. Then the proteins of the capsid are produced and the pre-genome is encapsulated together with the polymerase, with the reverse transcriptase, of the capsid are produced and the pre-genome is encapsulated together with the polymerase, with the reverse transcriptase, and the pre-genome is encapsulated together with the polymerase, with the reverse transcriptase, that once inside the virion produces double-strand DNA genome from the RNA positive. Produces double-strand DNA genome from the RNA positive. And in fact, if you observe, it happens the opposite than in the case of the retrovirus. The reverse transcriptase activity, it was the first activity in the cell cycle in the case of the retrovirus, and it is the last activity in the case of the epagnavirus. Now that we understand all the different RNA virus, now we are able also to understand better this table with all the different types of RNA genomes. Positive strand, negative, segmented, non-segmented, double-strand RNA, ambisense, undiploid RNAs with retroviral activity. And we have seen from all of them how they produce the mRNAs and how are the products, the protein products that they produce. But there's one thing that we haven't talked about, and is what is related to the third and the fourth column, and is if the virus take or wear a transcriptase in their virions by one side, and by the other side the infectivity of the genome. What you can observe is that only positive strand RNA virus without reverse transcriptase activity, only those do not need to take the transcriptase, the protein transcriptase with them. I mean, of course, they have to have a gene that codifies for this for this RNA polymerase dependent on RNA, but they do not have to take it with them as a protein, okay? But the rest of the RNA virus, they need to take the protein with them inside the virion, a part of the gene that codifies for that RNA polymerase. Only positive strand RNA virus without reverse transcriptase activity, only those do not, and by the other side the infectivity of the genome. What you can observe is that only positive strand RNA virus without reverse transcriptase activity to take the transcriptase, the protein transcriptase with them. I mean, of course, they have to rest of the RNA virus, they need to take the protein with them inside the virion, a part of the gene that codifies for that RNA polymerase. They have to wear it with the protein inside the virion, because the rest of the RNA genomes, the first activity that they have to perform is the transcription, and that's why they need an RNA polymerase dependent on RNA with them. Inside the virion is dependent on RNA with them. Is dependent on RNA with them. The genomes of RNA positive strand, the first activity that they do, picornavirus and coronavirus, in both cases, the first activity is the translation. So they are going to have their RNA polymerase dependent on RNA, even if they do not take it with them inside the virion. Okay? So, and that's what makes them their genome infective, because just the genome by itself will be translated and will produce the RNA polymerase dependent on RNA. Positive strand, the first activity that they do, picornavirus and coronavirus, in both cases, the first activity is the translation. So they are going to have their RNA polymerase dependent on RNA, even if they do not take it with them inside the virion. Genome infective, because just the genome by itself will be translated and will produce the RNA polymerase dependent and in fact this is the case for the positive RNAs, that they are infective. Here you see in this figure upside to the left how for example, this poliovirus, that is apicoronavirus with positive strand RNA genome, the poliovirus we can infect with the virus cell culture, okay, and produce new viral particles. But if we just take the genome without the rest of the virion, we can transfect with the genome, and this genome will produce new viral particles. And if we transform this genome in a cDNA, and we infect the virus with this cDNA, we will be able to produce new viral particles. And if from this cDNA we are able in vitro to synthesize RNA and transfect again with this RNA, we will be able to produce new viral particles. And this is so because the first activity that this virus, this genome wants inside the cells will do, it will be to be translated and to produce their own proteins. So this genome doesn't need anything else. And what happens with the other type of the other RNA genomes, the negative, the double strand RNA, ambisense, retroviral, etc. What happens is that they need, in order to infect, as the first thing, for example, this poliovirus, that is apicoronavirus with positive strand RNA genome, es muy interesante poder generar estos clones infecciosos. To do is to be transcribed, they need an RNA polymerase to produce their own proteins. So this genome doesn't need anything else. Their RNA genomes, in fact, as the first thing they have to do is to. And what happens with the other type of the other RNA genomes, the negative, the double strand RNA, ambisense, retroviral, etc. What happens is that they need, in order to infect, as the first thing they have to do is to be transcribed, they need an RNA polymerase dependent on RNA. Plus, fact, as the first thing they have to do is to be transcribed, they need an RNA polymerase dependent on RNA. And to get that, the only possibility to produce new viral particles is it would be just with a contransfection of their genome plus a plasmid of expression that includes their own RNA polymerase dependent on RNA. With this co-transfection, they will be able to produce new viral particles. Function of viral proteins for RNA. With this co-transfection, they will be able to produce new viral particles. And this is important to obtain infectious clones, because in this way we can perform experiments in the laboratory just to know the function of viral proteins. For example, let's say that we want to know what is a certain protein, viral protein for, what is their function. The only thing we have to do is just to transform the genome in a cDNA to mutate this specific gene for a protein. They will be able to produce new viral particles. Just to know the function of viral proteins, is their function. The only thing we have to do is just to transform the genome in a cDNA to mutate this specific gene for a protein. Clones, because in this way we can have to do is just to transform this specific gene for a protein, and then we will transfect cells and we will observe what happened with this virus, if it has any abnormality or not. So it has any abnormality or not. So it is a nice tool to study virus. These are called techniques or inverse genetics or reverse genetics, to transform the genome in a cDNA mutated, and in this way to see what a certain viral protein is for, what is the function, okay, or a certain protein domain. In the case of a positive strand RNA virus, we can do it directly just by mutating the genome in the form of cDNA and transfecting it. And in the case of negative strand RNA or double strand RNA or RNA with reverse transcriptase activity, what we should do is, apart from transfecting the genome, we would have to co-transfect with the RNA polymerase dependent on RNA. I turn back again to this table because in the case of retrovirus, there's an intermediate, that is the provirus, in the form of double-strand DNA, that this intermediate is infected. So retrovirus genome would be infected only in the form of provirus, I mean in the form of double-strand DNA. And just to finish this unit, I would like to talk about the concept of genomes, defective genomes that still they can replicate. And for that, just observe this figure. At the top, we have a wild-type genome that produces a wild-type virion. Okay, so that's fine. But sometimes in the replication process, you have mutant genomes that have some part deleted, and these genomes will not be able to produce wild-type virions. But sometimes what we observe in some virus is that we have a complete virion but with a defective genome. And how this virus is that we have a complete virion but with a defective genome. And how this can happen? This only can happen when the infection is with MOIs high in such a way that several viruses are infecting the host cell. And these genomes will not be able to produce wild-type virions. But sometimes what we observe in some virus is that we have a complete virion but with a defective genome. And how this can happen? This only can happen when the infection is with MOIs high in such a way that several viruses are infecting the host cell. And in this case, one wild-type virus may be the helper virus of the deleted genome. Happen. This only can happen when the infection is with MOIs high in such a way that several viruses are infecting the host cell. And in this case, one wild-type virus may be the helper virus of the deleted genome, and in such a way that the proteins of the virion are produced by the wild-type genome, even though we observe a defective genome in the virion, and in such a way that the proteins of the virion are produced by the wild-type genome, even though we observe And this is it. We finished Unit 5, and after me, Professor Tony Villaverde will give the lessons 6 and 7, talking about emergent viruses. And I think that in 1 month or so I will return again.
Evet. Arkadaşlar, izlediğiniz için çok teşekkürler. Şimdi kısa bir parça paylaştım. Devamını, aslında bir önceki canlı yayında çalıştığım kısmın devamını paylaştım sizlerle. Ve ondan sonra da koronavirüsle ilgili bir part var. Onu da çalışmaya çalışacağım. Yani hepsini böyle sırayla, finalim yaklaştığı için yapmaya çalışacağım. Belki viroloji için de işinize yarar. Burada İspanya'da aldığım dersler bunlar. Hocalarım gerçekten iyiler yani. Aynı zamanda bunu da paylaşmak istedim sizlerle. Belki bu notlar da işinize yarayabilir. İzlediğiniz için çok teşekkürler. Hepinize başarılar diliyorum. Motivasyonunuzu asla kaybetmeyin. Bir yanınızda suyunuz da olsun. Arada suyu da için. İzlediğiniz için sağ olun. İzlerken lütfen sizden bir ricam olacak. İzlerken videodan sesi kısmayın. Çünkü direkt laptopun sesini kısarsanız belki ne bileyim yan sekmede Facebook açık olur, bir şey olur. Oradan bir bildirim gelir, kafanız oraya gider ve aynı zamanda bak telefonum nerede bilmiyorum. Telefonunuzu da kapatın. Bu ikisi gerçekten insanı çok motive ediyor. Abone olmayı unutmayın. Devamı gelecek. Eğer istekleriniz varsa yorumda atabilirsiniz. Herkese çok teşekkürler. Çalışacak bir sürü konu var. Ara vermeden çalışacağız maalesef. Evet, herkese başarılar. Görüşürüz.